We are at the top of the hour, and today we have Dr. Peter van Endert. He's an MD PhD coming to us from Institut Necker-Enfants Malades in Paris. And the title of his talk is Insulin-degrading enzyme at the interface between beta cell resilience and autoimmune aggression. Just a little bit about him. He's a professor of immunology at University of Paris-Dakar, head of Department of Immunology, Microbiology, and Hematology at Institut Necker-Enfants Malades, INEM. And this is a public research center with 15 teams and about 250 workers, and he's head of the team, which is antigen presentation by MHC molecules, mechanism, and impact of toll-like receptors at INEM. And his primary research is focusing on different aspects of antigen presentation, cell biology, but he also studies cellular autoimmunity and type 1 diabetes. He's made major contributions to understanding the function and specificity of the TAP peptide transporters and discovered the role of ERAP2 and IRAP aminopeptidases in endogenous and exogenous antigen presentation by MHC class 1 molecules. We're really interested to talk with him today about his recent paper, Pancreatic Islet Stress Induced by Insulin-Degrading Enzyme Deficiency Promotes Islet Regeneration and Protection from Autoimmune Diabetes, which came out as a preprint in July 2023. I will put that DOI in the chat. But he's also going to share some unpublished work, so we're very interested to hear what he has to say. Welcome, Peter. Thank you so much for joining us.
Okay. Yeah. Hello, those who are online or those who will be online. So, yeah, and Monica, thank you very much for inviting me to present our data. So, if you have looked at the preprint, actually, there is some new data and some also was modified, obviously, since last July. Actually, this paper is in revision actually presently, but I'll also present you data from the second paper that is not presently submitted. So what I'm going to do is, I've been interested in this enzyme, insulin-degrading enzyme, for many, many years, for like 8 or 10 years already. And I'll tell you why that's the case. And it turns out to be a very complex story. And so today I'm going to highlight just a few aspects of what we found. And I can tell you right away, actually, the data we have that probably ask more questions than they answer. But so it's sort of an ongoing research. And personally, I think I'm going to be working on this enzyme for some more time. So I start with a few introductory slides, if that's okay. So I probably don't have to say much about type 1, type 2 diabetes, but I just wanted to highlight first that beyond the cliche view that type 1 is a disease where you have autoreactive T cells that destroying beta cells, and type 2 is a metabolic disease where you have sort of excessive demand. Beyond that, I think things are much less clear-cut these days. For example, you have inflammation in type 2 diabetes, subtypes of type 2 where macrophages, for example, inflammatory macrophages play an important role. The unfolded protein response, so protein homeostasis, are known to play a role in both types of diabetes. The beta cell is not only a passive victim now considered in type 1, but also sort of can resist to different extents to the autoimmune attack and so forth. So, and I'm mentioning this because what I'm going to show you actually highlights, the insulin-degrading enzymes plays a role in different contexts, both in the autoimmune attack and in the protein homeostasis. That's sort of the main messages I want to give you. Okay, insulin-degrading enzyme. So what protein is that for those who are not really very familiar with it? So it's a highly conserved in evolution. It's expressed in all cells. Obviously, it has a high affinity for insulin. That's why it has its name. But actually, what role it really plays in metabolism of insulin is not very clear, because when you have a knockout mouse, it doesn't have much of an hyperinsulinemia. It has an impaired glucose tolerance, but you can induce by simply injecting an inhibitor in a mouse. You get that immediately, this glucose tolerance that impaired. It's a particular protease because it prefers small substrates. It has genetic association with type 2, maybe also type 1 diabetes. I'm an antigen presentation person, so I first started to be interested in the role of insulin-degrading enzyme in antigen presentation by MHC class 1 molecules, and it doesn't play a broad role in that context. And what's important in the context of what I'm going to show you is that there has been speculation, or actually some data that can be interpreted as indicating that insulin-degrading enzyme plays a role in protein homeostasis in various cell types, in a number of cell types. Also, it might regulate the ubiquity in proteasome system. So there's a lot of complex work on that. Don't have the time to really discuss this. So insulin, you probably all know. I mean, insulin not only, of course, is that's the subject of that's what is produced by beta cells, but it's also, at least in the model, in the non-obese diabetic mouse model of type 1 diabetes, it's the key origin. So, essentially, one could say one self-antigen rules them all. Insulin is the only beta cell antigen that is required as a target of autoimmunity to trigger the disease. So if you kill essentially recognition of insulin by key CD8 and CD4 T cells, that's what George Eisenbart showed at the time, you don't get any diabetes at all. And so, and obviously insulin also is metabolic or challenge for protein homeostasis of beta cells, because it's produced in huge amounts, humongous amounts, like I have the numbers here on top of this slide. And because of this, protein homeostasis in beta cells is very important in the disease pathogenesis of type 1 and type 2. I think I probably don't have to present the non-obese diabetic mouse, right? It's sometimes it's debated how good of a model it is for human type 1 diabetes. Certainly, it's the best model that's around. If you compare to other autoimmune diseases like multiple sclerosis, it's a spontaneous disease, which is sort of rare to find. In fact, it shares molecular features with human class 2 molecules, for example. The older antigens are pretty much the same. So it's a really pretty good model. So the first set of data I'm going to discuss with you sort of response to that question. That was my initial question working on this enzyme. So I wondered, as an antigen presentation person, and considering the high affinity of IDE, let's say insulin-degrading enzyme, IDE for insulin, is it involved in antigen presentation of insulin? And that could, of course, affect two things: recognition of beta cells by autoreactive T cells, or also T cell education in the thymus, because we do know that insulin is, of course, expressed in the thymus. And so I'm going to show you just a few slides about this. That is the second paper that's not somewhere online. So we obtained an ID in knockout mouse. It's constitutive knockout, right? So it's knocked out everywhere. And some of what I'm going to present you, data obtained with a knockout mouse, probably has a background that is not only limited to functions in beta cells. I'm just going to say that, but that's another topic. So we backcrossed the mouse that came on the B6 background, of course, to the NOD strain, and looked at diabetes incidence. And the mouse is not entirely, but pretty much protected. Male don't get any, and female mice get very little diabetes. Also, we see that we look at infiltration, islet infiltration. We first have lots of normal infiltration, but then the score goes down, which is very unusual in the NOD model, right? That the mouse gets older, the sort of recedes the islet inflammation. We looked at T cells in peripheral blood, and so we used the two, let's say, most prominent CD8 T cell epitopes that are used in the NOD mouse model. One is this insulin epitope that's going to be important in the following. It's the peptide B1523. It's very immunodominant. And the other one that is even more immunodominant is this IGRP epitope 26214. Perry Santa Maria has done a lot on this. And so first thing we see, we have, to make it short, we have less autoactivity to both of these. When we transfer diabetogenic splenic T cells to an NOD skid mouse, we see that we have, and that's the red track here, we have a lower number of or lower activity of diabetic splenocytes in the knockout mouse. Okay, but however, when we do co-transfers between regulatory splenocytes and diabetogenic splenocytes, we don't see any difference whether the regulatory T cells come from the knockout mouse or the wild type mouse. Okay, so less diabetogenic activity, less autoreactive CD8 T cells in the blood, but no difference in regulatory T cells. So this is sort of busy slide, but essentially what we did here, just checked how is actually presented or how is actually processed and presented by a beta cell, this key, very well characterized CD8 T cell epitope of insulin. So how is it made? Okay, what are the proteases involved essentially? So to go quickly through it, what we show here, if you look at the endogenous pathway, so here it's the beta cell itself that makes the epitope. We see that it is made, at least in part, by what we call the constitutive proteasome. So that's the proteasome that's not in an inflammatory cell. That is sort of surprising because there were some reports that that would be the immunoproteasome. However, when we do a cross presentation, so here we sort of take beta cells, we get them killed and feed them to dendritic cells, then it's the immunoproteasome that does the job. Not surprising. What's most important is we looked at how this key epitope is presented by beta cells of mice that are deleted for insulin-degrading enzyme. And what we see here overall, although it's variable, we have a decreased presentation of the epitope. So insulin-degrading enzyme seems to be involved in making this epitope. Here are a few more things. When we knock out these aminopeptidase that actually Monica mentioned that I've been developed, been involved in also discovering, we see actually we have a knockout for this aminopeptidase, we have better presentation. So this aminopeptidase actually makes it, sort of destroys it partly. And here to the right, it's something interesting. So when we stress the mouse, then we essentially inject it with tunicamycin, not something very nice to do. We have increased presentation of the epitope. So that is probably not totally unexpected. Beta cell stress increases antigen presentation. It's not the first time that is seen. That has been shown for other epitopes, at least for one other epitope. Some more data, and here particularly, if you look at the left side, we also did a classic kill assay with chromium labeling of beta cells. And what we see here is the knockout mouse are much less killed than the wild type beta cells. So it's direct kill of beta cells. And so that confirming what we've seen in the previous slide, which was actually interferon gamma production by T cells. And this is another experiment that sort of answers how important is it actually for inducing diabetes, making diabetes in the mouse. So what we did here, we produced chimeras. So we irradiated mice and then gave them bone marrow stem cell grafts. And these grafts came from two mouse strains that expressed transgenic T cell receptors. So the 8.3 mouse expresses a T cell receptor. You probably all know that it recognizes the IGRP epitope. And the G9C8 mouse expresses a T cell receptor that recognizes the insulin epitope, 15 to 23. Okay, we use a lot these G9C8 T cells from Susan Wong in Cardiff. I think it's very easy to see here. For the 8.3, so the IGRP, the delay to get diabetes is completely identical, no difference. But the mice, the knockout mice that are the recipients here and that are of the chimeras, they get much later and much less diabetes. So that corresponds to what we've seen in the in vitro experiment. So we have protection against insulin recognition by insulin-degrading enzyme knockout. And so that's actually the first part, or actually the core of it. That's the paper that's not online. And so here again is the summary of that. So we have less autoreactive T cells, And we can conclude that we have a processing and presentation pathway by beta cells where insulin-degrading enzyme plays a role next to the proteasome. And so we have this protection that at least partly is probably due to a failure of efficiently activate CD8 T cells recognizing insulin. So that's the first story I wanted to tell you, first conclusion. Now, considering that IDE has, of course, a high affinity for insulin, we thought, well, maybe it's also involved in maintaining protein homeostasis in beta cells. For example, IDE could be involved in sort of getting rid of excess insulin in the beta cells or proinsulin. And so that could have an impact on the resilience of beta cells to, for example, if you have an immune challenge or if you have a metabolic challenge. And I already mentioned to you that there's literature data that suggests that insulin, that IDE plays a role in protein homeostasis. So we have ourselves produced some data. And so here, this is actually data from a collaboration years ago. Here we looked at yeast cells. So SPOM data. SPOM, although it's actually obviously very far from humans or mice, it expresses proteases but highly similar. It's like 45% identical to the human enzyme. And what they did, our collaborators here, essentially they knocked out, which is very easy, in this fission yeast, they knocked out IDE and looked at how resistant the cells were to ER stress. And that is induced by either tunicamycin, so you have an accumulation of non-glycosylated proteins, or also you can add urea to the cells. So what you see that the cells become more resistant. And that is actually dependent on, to make it short, on mTOR, right? And that is, I mentioned it because it's going to be reproduced in what we see later. So protein homeostasis is obviously extremely important in beta cells because they have huge demand, they're exposed to huge demand of producing, of synthesizing and exporting insulin in vast amounts very rapidly. So beta cells are known to have constitutively activated what's called the unfolded protein response. And that's probably, you probably all know that it's this pathway where you where the cell limits the amount of protein that's produced, ejects part of it into the cytosol for degradation in the sort of ERAT pathway. It also upregulates production of chaperones so that protect proteins. So that's very important. So that's, of course, something that's extremely well known. I just want to show you here that not only in beta cells, but there is evidence also from us that IDE plays a role in protein homeostasis. For example, when you take here on the left side, these RPMI cells, this is a myeloma, okay? So it doesn't make insulin, but it makes antibodies, it makes immunoglobulins. And you inhibit it with epoxamycin, so proteasome inhibitor, and you then add to it an inhibitor of IDE, you see that you have much, much more cell death that you induce by this sort of, by adding IDE inhibition to proteasome inhibition. Now that's a little bit more indirect evidence is here on the right side. We have taken beta cells, so we have taken beta cells from mice that we have treated with carfilzomib, that's the proteasome inhibitor, tunicamycin again. Or here we compare with the two IDE knockout mouse, and we look at simply by qPCR expression of IDE. And we see that these two stressors, massive stressors, both upregulate substantially IDE expression very rapidly, which suggests that this is important if you have accumulation of unfolded proteins or if you have inhibition of the main system for getting rid of accumulated ER proteins, then the IDE sort of comes up to sort of help. Okay, so then now what about our knockout mice? And the results I show you that here first on the left side, here's we are looking at black 6 mice. Okay, so we also worked on those. IDE knockout black 6 mice, and we look at a sort of number of sort of key proteins by qPCR or genes by qPCR that are involved in the unfolded protein response. Well, and then simply you see immediately that we have an upregulation BIP. So the key indicator of the unfolded protein response. Also some XBP1 is upregulated. So we have what can be called a mild unfolded protein response at steady state. So we have not done anything else to the mice. This is sort of the steady state of the mice. I get later to looking at the same thing in the NOD mice, where we have actually a different story. Now, as you may know, there is published evidence that stress, beta cell stress, not only induces an adaptive response by, for example, upregulating chaperones, increasing the ER, so forth, but can also, at least as long as you have mild stress, can upregulate proliferation. So this is sort of summarized in this slide. And so we looked at that also. And what we find actually, and that is here, two different situations where we stress the cells to sort of, and where we also look at the steady state. So on the left side, we have stressed the NOD mice with tunicamycin. So again, we have this accumulation of unfolded non-glycosylated proteins. And the right side is a more physiologic stress, if you may. So we exposed B6 mice to a high fat diet for 6 weeks, I think. What we can see, and the knockout mice are always in red here, and you can see at steady state already, we have, this is, the readout is Ki-67. So we look at that by microscopy proliferation. So we have an upregulation at steady state, tunicamycin, again, we have much more Ki-67, and that we can actually, if we sort of counteract protein production by adding rapamycin, by treating mice with rapamycin at the same time, the effect is gone. So it's mTOR dependent. And we actually, as you remember, we have shown that before in the yeast, it's the same thing. And the same is the case if we give high fat diet. We also have, I can't really see that because I have the second. Also, yeah, there we have a clearly more Ki-67 in the knockout mice. Okay, you see, yeah, and so we also looked at this at steady state and in beta cells. So we did here, we did co-labeling with insulin, and in that case we didn't use Ki-67, but we use EDU labeling. And again, whether it's stressed B6 mouse or a steady state NOD mouse, we have more beta cell proliferation. Now, just a quick, and that is actually in the paper that you may have seen. This is sort of a view on the overall, let's say, state of glucose metabolism or beta cells in that context. And what we see is that we have a hypertrophy of the beta cell apparatus. So we have a higher number of islets. We have, at least here, this high glucose situation, we have a higher output of insulin, glucose-stimulated insulin secretion. We have also slightly pathologic glucose tolerance, the oral glucose tolerance test, which is consistent with the literature. And this here, IAPP, okay, is you will probably not know, it's a known substrate of IDE. So this is in the serum actually highly increased. There's a lot more of it. So I showed you proliferation, but also this beta cell hypertrophy. And so here is a view on what happens with this mouse, with the B6 mouse, if we expose it to a high fat diet for six weeks. And what you can see here, if you go through it, we see that the mouse will actually have a lot more weight gain than the wild type mouse. And the beta cells sort of start to wildly sort of overproduce everything. So they have actually huge capacity to output insulin, which we find in the serum, or we can extract it also from the islets. serum proinsulin is even more increased in the serum, which is sort of an indication of immature. The beta cells essentially don't cope with all the insulin or proinsulin that they produce, so that it actually gets into the serum. And despite all of this, the mouse, the knockout mouse, develops sort of type 2 diabetes, essentially becomes hyperglycemic. So, which is sort of an amplification of, if you may, of the steady state phenomenon that we have found. So essentially, we have a situation where, despite hypertrophy, we have a decompensation of metabolism, at least in a stress situation, be it metabolic stress or proteotoxic stress. And I wanted to add to this, which is actually something that we don't understand completely, why that's the case. Why do we have this hyperproduction of insulin? Why do we have this increased output at steady state or upon high fat diet? And why does the mouse anyway become diabetic? So, and these are the most recent results that we have that I wanted to show you, because so we have the paper actually in revision, where we show the data on the high fat diet, the proliferation, and the hyperglycemia that develops and so forth. And so one reviewer sort of suggested to us to look a little bit at the ultrastructure of beta cells. What we did, and the result was very surprising. So what you can see here is a view of beta cell, wild type beta cell. So you probably know all this. So you have the dense core here, and with the halo around. And here is a knockout cell. You see immediately it looks completely different. It's absolutely dramatic alteration of the insulin granules with this huge halo. And you can actually quantify that. So you have, well, first thing, we have the overall surface of beta cells is bigger, and that corresponds to the volume by conventional microscopy that we found bigger. What is increased is not the dense core, so the insulin, but and actually the number of granules per beta cell is decreased because the granules are just much bigger. But when you look at the ratio between the diameter or the surface of the halos relative to the beta cell, it is hugely increased. So this is a very recent result, and we don't really understand why it's the case. And just maybe not surprising, but we're sort of good company not understanding, because there's this paper here on PNAS. It's published 2019 from actually real beta cell experts, and like not which I am not really, including Michele Solimena, for example, in Dresden, and they looked at beta cells in a mouse with a deletion for this protein, ATP6, AP2. Maybe the name already tells you that this is a protein that's important in assembly of the VATPase. So the VATPase, this lysosomal enzyme, important for mTOR regulation, for acidification of lysosomes. And so they went through the things that you do when you see that you have something deleted that is sort of part of the VATPase. And they found that they had sort of excessive autophagy, so elimination of insulin granules, which is not what we actually see. And they had normal pH. The VATPase seemed to function normally. So in the end, they concluded, and this is actually very well done, PNS paper. They said, well, apparently something is wrong about protein homeostasis. Okay, and that's obviously what we also see, but we also believe what's happening here. But what exactly causes this phenotype with these huge halos? We have no idea at this point. And again, even these experts here actually were not much smarter about it. So here's second conclusions, and they're going to be a third set of conclusions, and I'm going to be done. So IDE is induced if you have stress, be it proteotoxic or metabolics, and it contributes to protein homeostasis. It induces a mild islet cell stress, UPR already at the steady state. that is associated with better cell proliferation, both at the steady state and in the stress situations. And that's dependent on mTOR and what I didn't actually comment here, but that was one slide, ERF to alter dephosphorylation, because we had an inhibitor we used. And in the INOD mouse, of course, the proliferation may contribute to protection from diabetes, in addition to, let's say, the reduced recognition of beta cells by insulin-specific CD8 T cells. And I had showed you the hypertrophy, increased GSIS, and so forth, and then this dramatic vacuolation that really remains to be understood what it means. Okay, so last part. It's here we get back to the NOD mouse, but this time we're not talking about T cell recognition of beta cell antigens, but we again look at the stress situation. So here on the left side, I showed you before, this is the B6 mouse at steady state, BIP upregulation. It's the classic UPR thing. But in the NOD mouse, it's a completely different story, very surprising. There's no BIP upregulation, but the REC2 is upregulated. Now REC2, I'm going to tell you in a second what it is if you don't remember. REC2 is, and this is just to show that we did a proteomic analysis of islets, and we also found REC2 also induced in the knockout mouse. It's not because IDE would actually digest REC2, it's actually too big, so it was unlikely anyway. So what is REC2? REC2 is a protein that is a relatively small protein, but too big for IDE to degrade. It's normally expressed already in exocrine pancreatic cells, but not at all in beta cells. In beta cells, it's really exclusively expressed when they regenerate. And it has some interesting functions in the context of our study. So it attenuates the unfolded protein response and apoptosis. It can induce BIP, and it has an anti-inflammatory effect. And in fact, there was a number of years ago, people were interested in REC because it's actually so secreted. It can be produced, of course, and they showed that it had sort of protective for beta cells, in vitro at least. Now, what's interesting about this in this context, and that's another thing that we are very sure about the phenomenon, but we don't really know why that's the case. We have found that this REC2 induction that's specific for the NOD again is entirely linked to autoimmune aggression of the beta cells. And so here, how do we show that? For example, in female NOD mice, because they get diabetes much more than males, we have a higher rate of, so if you look at the factor of increase, it's much higher in females than in males, that they get much less aggressive, of course. Autoimmunity. When we, and I'm not sure here, but I'm showing here, when we pre-incubate the beta cells overnight, and you may know that actually the immune cells they leave the islets. You can see that on the microscope. Just go out. Macrophages, T cells, everybody goes, just leaves them. And then we look the next morning to the same PCR cells, gone completely. It's for the BIP induction in the B6 is not at all the case. That's an intrinsic story. But here, it depends on the immune cells. Another thing that we did, yes, we also looked at IDE RAG2 double knockout cells, or islets, sorry. So these don't have any immune infiltration. There is no REC2 induction whatsoever. So it's another argument for that. It's linked to inflammation. And another fact, when we look at what happens, when we look at by qPCR what happens when we expose the NOD islets or the mice, and then take out the beta cells and do the qPCR after these massive stress that we've used before, like tunicamycin or whatever, proteasome inhibition, there's no REC2 induction at all, a little bit contrary to expectation. So the REC2 induction is not triggered by massive tunicamycin sort of type stress, but it's specific answer in the NOD to autoimmune stress, the T cell essentially, attacking the beta cell. Then they start making this, which is I find interesting. I would like to know how that works. So these are the third set of conclusions. So as I insisted a lot, in the NOD, contrary to B6, the ID deficiency induces REC2. And this is only done by immune stress. And that, of course, could also contribute to protection of IDE, NOD mouse from autoimmune diabetes. So we have like three things that could predict: less recognition of insulin presented by beta cells, or also by cross-presentation maybe, the proliferation, and REC2. So a lot of things that could actually protect the NOD mouse from diabetes. And
Peter, can
I
just was waiting for a good
Yeah.
pause here. There's a question from
Yeah,
Yang Dai. His question is the IDE knockout
done
specific?
anyway.
Okay. Well, he had a quick question. Do you want to, he can unmute himself, but I can also ask, is the IDE knockout specific for beta cells? Question
No,
one.
it's not. No, it's
Okay.
not. And that's why I said what in the very beginning. It's constitutive. I mean, we have the conditional one in principle. We have it in the lab, but it's way complicated. So we didn't have the time to look at that.
Okay. And the second, he has three questions. Second one is IDE substrates in addition to insulin.
Yeah, there's a number of substrates that are known. Again, they are all small substrates. I mean, an important substrate in that context is IAPP. IAPP is very well known, very efficiently degrade. Glucagon can also be processed by insulin, and a number of other hormones can be produced, but it's all small or mainly sort of hormone types.
Interesting. And then on your suggestion of one antigen rules all, does this antigen remain as the driver throughout the T1D course?
Yeah, probably not. I mean, I would think that it's, I don't know whether anybody has, no, I think it hasn't been done. Like, for example, the experiment to answer really the question conclusively would be that you have sort of an inducible substitution. It's the substitution of insulin, position 16 of insulin kills the recognition by CD8, CD4 T cells of insulin. That's the George Eisenbart would probably showed. So the idea would be you can induce that somehow along the line and see what's the result. I would think the result of this, in fact, very complicated experiment would be that just you have no effect on autoimmunity, because it's known that you have probably the first pretty much CD8 T cells, at least, that you see in the NOD mouse are the 1523 insulin B chain pipettes, but then you have IGRP takes over. And IGRP, CD8 T cells, they're much more frequent at some point. And I don't think, it's this epitope spreading story where you don't need the initiator anymore at some point, I would assume.
That's so interesting. We featured in our weekly digital newsletter, the WEIR Week in Review, which is specific to type 1 diabetes. Last week, a preprint from Joachim Storling, he's in Denmark, Copenhagen, titled Beta Cell Pyroptosis, a Burning Flame in Type 1 Diabetes, where they postulate whether or not it's really pyroptosis versus apoptosis, and that is kind of tying into this idea of spreading. So I wondered if you wanted to comment on that. Maybe you haven't even seen it because it's pretty new paper. But how might this construct that you're discussing fit into a pyroptosis model?
I don't know. There are many really surprising things, I think, in beta cell physiology and how you can intervene with type 1 in the NOD model. For example, you probably have seen that paper. Yeah, I mentioned the unfolded protein response, right? So there is this paper, I think it's cell metabolism or something. They knocked out ATF6, and which is one of the three branches, right? ATF6, and I think also IRA1 did the same job. What happens is the beta cells become senescent, early senescent, and you get some M2 macrophages that sort of gobble up the senescent beta cells. And the result is that the mouse is pretty much protected from diabetes. I think you need to understand that. It's really amazing. I mean, it's sort of anti-intuitive. The other story is you sort of, also maybe cell metabolism paper or something, you sort of diminish the volume of beta cells, take away half of the pancreas, something like this. It's also protective. So we need to understand that. So yeah.
It's very interesting. Then, oh, sorry, I
Yeah.
just also wanted to ask, obviously IDE is a metalloprotease, and I believe zinc is the metal, right? So
Right.
it's kind of interesting, right, that zinc A transporter is one of the autoantibodies. Do you feel like there's, or do you think that there's any sort of connection there? Is that like some kind of, shut off valve or something?
Yeah, you see the ZNT8, of course, is I think it's associated with secretory vesicles, right? But you need for the insulin crystals, you need zinc, right, in the secretory granules. Now the thing is that actually IDE is not in the secretory granules, right? And so, which is also sort of because I've been collaborating with Michele Solimena also.
Yeah, we know him
in the context.
He's spoken to us.
Yeah. And so the question is also why Michele thinks that you have an accumulation of old granules, right? He's a specialist that distinguishes young and old granules, and he thinks that you have an accumulation of old granules. Now, how that works is really also a tricky question, because IDE is not going to be important in directly as a protease in degrading a secretory granule. Can't do that. I mean, it's not in there, and how would you get access? It can be worked in an ARAT pathway. Maybe that's the class 1 addition presentation. But there seems to be something more happening, right, in the beta cell. So somehow, and I think that's why we have this dramatic vacuolization, IDE has an effect on what happens to the secretory granules. So it means you're somehow talking about an effect on crenophagy or autophagy. And that is probably not a direct proteolytic role of IDE, but something indirect that somehow regulates protein homeostasis. And then you come, I could continue forever. It gets ever more complicated. You have, VA-ATPases, mTOR is regulated, and so forth and so forth.
Is there any kind of like imbalance though, in terms of like the global sink of zinc that's in the beta cell? And that's kind of what I was thinking about when I asked the question is like, if IDE is still, alive and well and, working, it needs zinc. So it has access to zinc, but is the zinc A transporter still working properly as well? Is there some like paucity of zinc over at the zinc transporter site? Is there some kind of, stealing from Peter to pay Paul kind of thing going on with the zinc? It's just just a real thought experiment, but.
Yeah, I see what you mean. Yeah, I don't have an answer. Of course, I wouldn't. I think the amount of zinc that's very low that your protease needs, something like this. But I don't know. I would think that you need much more zinc for the
transporter
for making the dense cores that look actually normal. I mean, the surface is normal of the dense cores, and so I would think that it should be enough zinc also for IDE, or that shouldn't make much difference whether IDE consumes or not. See, but I don't know.
Yeah. Anyway, it's sort of an out there question. Here's one more question. Does the driver antigen have to be derived from beta cells? We have some antigens derived from stem cells. This is from Yang Dai again.
Well, the driver, I mean, essentially you're getting to the very big question, like what is really triggering the autoimmune attack, beta cells? I mean, what's at the beginning? And there is this. I was sort of sorting through my papers today. I got this study from Annette Ziegler some time ago. It says that you have they follow like 1,000 children with whatever high risk, And they see a peak of hyperglycemia 2 months before they get their first autoantibodies.
Mm.
So really, and so to get back to the question, of course it could be. I mean, people would think about an infectious antigen, and then we have mimicry or direct inflammatory effects. But stem cell antigens, no, I'm not aware of that. So I don't know what is the background of the question, but I'm not. Many antigens are known, and there is the idea of post-translational modifications, so forth, in the beta cells. But I'm not aware of any stem cell antigens. So what's the context of the question? Can I ask that?
Yang, do you
Yeah.
want to unmute and discuss?
Yeah. Hi, Peter. This is Yang
Hi.
from San Diego.
Yeah.
So we have been working on GAT65 for a long time, and the processing issues. That's why I got interested in your presentation. And after Eli passed away, I was thinking what kind of antigens could be the drivers, which he believes strongly. So I ended studying insulinoma cells, mesenchymal islet stem cells, and trying to figure out what the protein antigens from those cells in NOD mice. We ended in endogenous retroviral proteins.
Okay.
So we have published that. We got a very slow. We're targeting one of the endogenous retroviral antigens, GAG, which is one letter different from GAT. It's very interesting antigen because this is retroviral antigen, so there's a plenty viral characters.
Okay.
And that's why I bring up
Okay.
whatever environmental factors could be a side effect of targeting the endogenous retroviral antigens, which become the target as the primary driver antigen. So that's how I bring this question up. Thank you for your presentation.
I see. Okay, interesting. Thank you. I know there was a story maybe many years ago. It was from a Swiss group, right? I mean, I'm probably, you need to be as old as I am probably to remember this. I know they looked at like, I think it was beta cells from like 10 people or 20 people, and found activation of endogenous retroviruses. But then there was from Geneva, I think, the study at the time. But and it then turned out to be some technical issues. But certainly, I mean, endogenous retrovirus could play a role. So is there any evidence that you would have is that any evidence that you have sort of activation event that would make them visible?
Initially, we use just a T cell readout, which is a very degenerate readout. We tested some clones and some functional readouts by tolerance induction to the candidate, and of course interferon gamma secretion T cell response. So those data is weak, but so far we knocked down by RNAi of the target, while knockdown is not 100% by shRNA. So it is a partial knockdown, but it gave us confidence this antigen can affect T cell response very strongly than all the antigen we know for sure is better than GAT65. So this retroviral antigen does affect T cell response some way, somehow. That's the conclusion from the T cell readout part, which is difficult one. Recently, we start monitoring deep sequencing the sequence of the retrovirus. Very exciting though in mouse where the paper is under review.
Okay.
It's a complicated antigen. So we need to really sequencing them out. Thousands different sequences. So as you mentioned, the Swiss group in 97 published in Cell paper.
Yeah, right.
It's very difficult. It's very easy to make mistake because the contamination of the genome DNA, which they didn't exclude that, of course. That's how we are moving here in San Diego.
Okay, great. I think they had an issue with were using fresh cells on one side and like shipped or frozen cells on the other side, right? Was something like that. It was the Swiss group.
Yeah, is the genome DNA contamination the major issue?
Okay. Yeah.
Because you got many endogenous retrovirus in the genome, of course.
Yeah. Okay, great.
Well, that's really interesting, Yang, and we'll be in touch to sort of, request that you can fully flesh out this story once it's accepted. And so it really sounds interesting. So we'll be reaching out to you soon.
Of course.
And
yeah,
great. And Peter, thank you so much for this really interesting talk. It's fascinating, and I love how you are collaborating with Michele and others in Europe, closely really scrutinizing the cell biology of this complex disease. It's fantastic.
Thank you, Monica. Yeah,
thank you,
it was a pleasure.